Hello everyone, welcome to JK Biology. In the previous video, we already discussed about anterior and posterior axis formation in Drosophila. In this video, we are going to discuss about dorsal ventral polarity in Drosophila. Already we know about which one is the anterior region and which one is the posterior region. We'll see now which one is called as the posterior, which one is called as the dorsal and which one is called as the ventral. This is anterior region. Anterior region means head region, and this is the posterior region. Posterior region means abdominal end region. Head region is called as the anterior region, abdominal end region is called as the posterior region, and back of the fly is called as the dorsal. Back of fly is the dorsal region, and belly portion of fly is called, belly region is called as the ventral. That is called ventral region. Is it clear about the anterior, posterior, dorsal and ventral regions? Now we'll see how this dorsal ventral polarity is formed and when this is formed. Dorsal ventral polarity forms, dorsal ventral polarity forms, it forms before fertilization. Before fertilization. First we'll see how the dorsal region is formed. We'll draw our diagram of the egg chamber. This is oocyte nucleus and are the nerve cells, and these oocyte and nerve cells are covered by the ovarian follicle cells. Are covered by ovarian follicle cells. Now here, this is the oocyte, nerve cells, follicle cells, ovarian follicle cells. As the oocyte grows, its volume increases. Oocyte volume will increases, and nucleus of oocyte moves to dorsal anterior region. Dorsal anterior region. Here is the nucleus. Once the oocyte grows, its volume increases, and nucleus of oocyte, this nucleus will move towards the dorsal anterior region. Here, this is the nucleus of the oocyte. Now later what happens? Nucleus of oocyte produces one mRNA that is called as the gherkin mRNA. It produces gherkin mRNA. See here, this nucleus will produce the gherkin mRNA. This is the gherkin mRNA. Later, this gherkin mRNA is translated into gherkin protein, that is translated into gherkin protein. This gherkin mRNA is translated into gherkin protein. Later, gherkin protein is received by follicle cells which are closest to oocyte nucleus. That is, this gherkin protein will be received by the follicle cells which are nearer to the oocyte nucleus. And inside this follicle cells, there will be presence of the one of the receptor. That receptor is the torpedo receptor. This is inside follicle cells. Receptor is present. That receptor is torpedo receptor. That is the torpedo receptor. Remember the name of the receptor, torpedo receptor. Now this gherkin protein binds to torpedo receptor which is present in the follicle cells. This binding will inhibit this binding of the gherkin protein to the torpedo receptor. This will inhibit the expression of another gene. That gene name is pipe gene. Remember, expression of the pipe gene is inhibited by the binding of the gherkin protein to the torpedo receptor. Due to the inhibition of pipe gene expression, the cells dorsalizes. The cells differentiate into dorsal cells. This is about the formation of the dorsal polarity. Is it clear? See the overview of this. Here, as the oocyte grows, that will produce the oocyte nucleus will produce the mRNA, that is the gherkin mRNA. Later, this gherkin mRNA is translated into gherkin protein, and this gherkin protein is received by the follicle cells which are nearer to the oocyte nucleus, and inside these follicle cells, the gherkin protein will bind to the torpedo receptor. It will bind to the torpedo receptor which are present in the follicle cells, and binding of this will inhibit the expression of the another gene. That gene is nothing but the pipe gene, and due to the inhibition of the pipe gene expression, these cells will differentiate into dorsal cells. Now we'll see the ventralization of the cells. In the ventral pollicle cells, there will be no Gherkin mRNA. The Gherkin mRNA is not entered into the ventral pollicle cells. So here, Gherkin mRNA is not entered into ventral pollicle cells. Due to absence of the Gherkin mRNA, there is no Gherkin protein. No Gherkin protein. So here there is no Gherkin mRNA and there is no Gherkin protein. No Gherkin protein. Absence of the Gherkin protein will lead to the expression of the, due to this absence of the Gherkin protein, pipe gene get expressed. Pipe gene get expressed in pollicle cells. Now, due to expression of the pipe gene, the pipe enzyme is synthesized. Pipe enzyme is synthesized. That is, here pipe enzyme is synthesized. That is, name of the pipe enzyme is pipe sulfotransferase enzyme. Pipe sulfotransferase enzyme. As the name suggests, this pipe enzyme will sulfonate the vitreline membrane proteins. See here, this pipe enzyme will sulfonate the vitreline membrane proteins. Here are the vitreline membrane proteins. These are sulfonated by vitreline membrane proteins are sulfonated by pipe enzyme. Later, sulfonated vitreline membrane proteins, they will bind to another protein that is called as the GD protein. Sulfonated vitreline membrane proteins bind to another protein that is GD protein, gastrulation defective protein. GD protein, that is gastrulation defective protein. This is the gastrulation defective protein. And later these two complex, that is vitreline membrane protein and GD protein, will bind to the another protein that is called as this snake protein. And this snake protein, firstly it will be in the inactive protein, that is vitreline membrane protein, GD protein will bind to another protein that is binds to snake protein. Snake protein, which is in the inactive form. This is in a inactive form. This snake protein will be cleaved by GD protein and it will become active snake protein. Now this become active snake protein. Is it clear? Wetland membrane protein and GD protein will bind to the snake protein, and initially this snake protein will be in inactive form. Later the snake protein will cleaved by the GD protein and vitelline membrane protein, and vitelline membrane protein and it will forms the active snake protein. See here, GD protein will cleave the snake protein and it becomes active. Now activated snake protein will binds to another protein that is called as the ester protein. Active snake protein. This active snake protein will binds to another protein that is ester protein. Ester protein, which is also in the inactive form, and this ester protein will be activated by, cleaved by snake protein. Snake protein will binds to ester protein and cleave it, and it becomes the active ester protein. Active ester protein. Active ester protein. Now the active ester protein will binds to another protein that is called as the sphagal protein. Sphagal protein. This is also in the inactive form. This sphagal protein becomes active by cleaved by ester protein. Ester protein. Activated ester protein binds to sphagal protein and it will cleave the sphagal protein and it becomes the active sphagal. Active sphagal. See here in the diagram. Firstly, what happened? The pipe enzyme will sulfonate the vitelline membrane proteins, and sulfonated vitelline membrane proteins will bind to GD protein, that is gastrulation defective protein. These two complex, that is the GD protein and sulfonated vitelline membrane proteins, will bind to next protein, that is the snake protein, and this snake protein will be cleaved by this complex GD protein and vitelline membrane proteins and becomes active. Later, activated snake protein will binds to another protein, that is the ester protein. Ester protein initially it was also in the inactive form. It is get activated by the cleavage by the snake protein. Snake protein will cleave the ester protein and make it active protein. Activated ester protein will binds to another protein, that is the sphagal protein. Sphagal protein is also initially in an inactive form and it will get activated by the cleavage by the ester protein. Ester protein will cleave the sphagal protein and make it active form. Now this activated sphagal protein will go and bind to the toll receptor which is present in the oocyte. This binding of the sphagal protein to the toll receptor will activate the protein kinase which is called as the pele, which is in association with the tube, another protein that is called as the tube. After the activation of this pele, it will phosphorylate this pele will phosphorylate the another protein, that is the cactus protein, which is in association with the dorsal. This is the cactus protein and this one is the dorsal protein. Once the pele activated, this will phosphorylate the cactus, that is this will leads to the separation of the cactus and dorsal protein. After the phosphorylation, this cactus protein will degrade and this protein, dorsal protein, becomes free. And now this dorsal protein will enter into the nuclei of this ventral follicle cell. This dorsal protein will enter into the nuclei of the ventral pollicle cells and they will get ventralization of the cells. See here what happened. Gherkin mRNA is not entered into the ventral pollicle cells. Hence, there is no Gherkin protein. Pipe gene, due to absence of the Gherkin protein, there will be pipe gene is get expressed in the pollicle cells. Due to expression of the pipe gene, there will produce the pipe enzyme is synthesized, and this pipe enzyme will sulfonate the vitelline membrane proteins. And sulfonated vitelline membrane proteins will bind to the GD protein, that is nothing but the gastrulation defective proteins. And these two complex, that is gastrulation defective protein and vitelline membrane proteins, will bind to another protein that is called as the snake protein. Snake protein, initially it is in an inactive form, later it is get activated by the cleavage, that is snake protein is cleaved by the GD protein and vitelline membrane protein and becomes active. Now activated snake protein will binds to the another protein that is called as the Easter protein. Easter protein is also in an inactive form. Easter protein is converted into active form by the cleavage by snake protein. Snake protein will cleave the Easter protein and make it active protein. Now, activated Easter protein will bind to another protein that is called as the spagel protein. Spagel protein is also in an inactive form. Later, it is get activated by the cleavage by Easter protein. Easter protein binds to spagel protein, cleave it and make it active spagel. Now, later what happens? Activated spagel protein, it will binds to toll receptor present in oocyte. Spagel protein will binds to the toll receptor, and this binding will activate the protein kinase. This binding activates protein kinase called Pele, which is in association with another protein, tube protein, which is in association with tube protein. This Pele leads to phosphorylation of cactus protein which is in association with dorsal protein, another protein that is the dorsal protein. After phosphorylation, cactus and dorsal protein get separate. Cactus protein degrade. And dorsal protein become free. Now this free dorsal protein enter into the ventral pollicle cells, enter into the nucleus of ventral pollicle cells, and there will occur ventilation of cells. Ventilation of cells. This is about dorsal ventral polarity in Drosophila. Thank you.